So this is part 2 of Chapter 17's PowerPoint lecture. And we were talking about how there are 3 different RNA polymerases, and they differ in where they actually do their transcription, so in terms of identifying which gene they're going to transcribe. So that really brings us to the question of how does the cell actually decide which gene gets read and transcribed? Because the latest estimate is that a human cell has about 21,000 different genes to choose from. And so sometimes some of those are constantly being transcribed, and others are only transcribed, for example, during embryonic development or during differentiation phases. So what is it that's going on that turns and controls the transcription of a gene? So first of all, to begin that discussion, we have to look at the structure of the actual strand of DNA. And what we have on the DNA just before the actual DNA sequences, nucleotide sequences that are the gene, before we get to that, there's a section of DNA that's called the promoter region. And it's actually a binding site that is located a little bit upstream from the gene. And it's a binding site for the RNA polymerase II. And the other thing that actually allows the RNA polymerase II to bind there are transcription factors, and we'll talk about those in a second. But this complex of transcription factors and RNA polymerase II actually is how a gene gets identified. Part of that promoter region is what's called a Tata box, which is essentially just a repeating TNA sequence of those different nucleotides. But they are crucial for forming what we end up calling an initiation complex. Another thing that's involved in eukaryotic initiation of gene expression is an enhancer region. And not all genes on every chromosome have an enhancer region, but some genes do, and particularly genes that get transcribed a lot have this enhancer region. And a lot of times it can actually be thousands of nucleotides upstream from the actually encoding gene. But it is a region that is a binding site for other proteins. So there are proteins that will actually bind to this site, and they enable then the transcription rate of the actual gene to be increased, and actually greatly increased. So it's a way to speed up transcription. So these enhancer regions, they're really actually learning more and more about them. But what they do know is when they are present and when they are bound, they increase the transcription rate a lot. So next, let's take a look at transcription factors. So transcription factors are a suite of different proteins. There are lots of them, which actually bind to the DNA. There are some that actually bind to the DNA. They're in the promoter region. There are others that actually bind to this initiation complex itself. And so these transcription factors, along with the RNA polymerase II, they have to be present first. And then the RNA polymerase II can then come along and bind to the DNA to begin transcription. And what is really thought is that these transcription factors, a lot of them are kind of like the on switch for transcription, and they actually get made or released as a result of some sort of hormone signal. And so we learned earlier about hormones as signal proteins, and also as steroids that actually work within the cell and oftentimes inside the nucleus. And these hormones are believed to play a big part in the release or production of transcription factors. And in that way, hormones are actually directly involved in the expression of the gene. So again, if you think of the fact that a gene is not going to get read, which means a protein is not going to get made unless we have these transcription factors. These transcription factors are the key really to the lock on the gene. And these transcription factors can be controlled by the presence or absence of hormones. This purple box at the bottom of this slide shows you what we consider to be the transcription initiation complex. So again, this is a completed assembly of transcription factors in the RNA polymerase II in a eukaryotic cell. This is a series of pictures showing the initiation of transcription. So transcription is, again, involves three things: initiation, elongation, and termination. So what we've been describing so far is just initiation. So you can see in that first top picture, we have a eukaryotic promoter region that begins with the ta-ta box, and then we have the starting point where the RNA polymerase can actually bind, and then we have transcription factors that bind to the DNA molecule, and those transcription factors then allow for the building of this initiation complex that includes the RNA polymerase II. During transcription, I want to restate that only one of the two DNA strands provides the template for the reading of the sequence of nucleotides, and that is called the template strand. And that template strand is always the same strand for a given gene. So it may alternate between the two different strands depending on which gene is being read, but a specific gene will only read one strand as the template strand. So now step 2 in transcription is elongation of the RNA strand. So as RNA polymerase moves along the DNA, it actually untwists the double helix 10 to 20 bases at a time. And it progresses at a rate of about 40 nucleotides per second in a eukaryotic cell. So RNA polymerase is not only able to bring in the new nucleotides for the growing RNA strand, but it's actually also able to unwind, untwist, and separate the double helix of the DNA so it can get to that template strand. And a gene can be transcribed simultaneously by several RNA polymerases. So this can be occurring over and over again as there are several RNA polymerases reading the same gene over and over again. And just like in replication, nucleotides are added to the 3' end of the growing RNA molecule. The last step of transcription is the termination, and the mechanisms for termination again are different in bacteria and eukaryotes. So in bacteria, it's very simple. The polymerase stops at the end of the terminator, and the messenger RNA can be translated without any further modification. So basically, in bacteria, the RNA polymerase will just stop at a sequence that's called the terminator sequence, and at that point the messenger RNA is finished. There's no more processing that has to occur. And for this reason, actually, translation of messenger RNA in a prokaryote can actually occur before transcription ends. So transcription can be going along, and translation can actually, the ribosomes can actually start translating that messenger RNA before it even gets to the terminator. because there's no processing needed of that messenger RNA. But in a eukaryote, RNA polymerase II will transcribe until it gets to the polyadenylation signal sequence. And that RNA transcript then gets released about 10 to 35 nucleotides past the polyadenylation sequence. And again, just to remind you what that polyadenylation sequence is remember we end up with the 5' cap and the 3' tail of our messenger RNA, and that tail portion is a series of nucleotides, and that is polyadenylation. So this is another picture showing the difference. In bacteria, the polymerase stops transcription at the end of the terminator, and then the messenger RNA can be translated. So that's being shown in this top one here, where the RNA is being released. But in eukaryotes, the RNA polymerase II will, boy, you're going to have to really zoom in to see this picture down here. But what this is showing here is this polyadenylation sequence. Actually, you can see it right there. A series of A's that are going to be made down here to be actually forming that 3' tail that we need. So I mentioned about 2 slides back there that in a bacterial cell or a prokaryote, the messenger RNA that is transcribed is ready to start translation. And this is all occurring within the cytoplasm of the bacteria. So the ribosomes are floating around and able to begin work on that messenger RNA right away. But in a eukaryotic cell, we have all of our transcription occurring within the nucleus. And what is first transcribed is really what we would call a pre-messenger RNA. It is not yet ready to do translation. It needs to be processed before it even leaves the nucleus and begins the process of translation. And part of the reason for this is eukaryotic genes have junk, which is kind of interesting. I think that idea of it being junk is kind of going by the wayside as we learn more and more about what's involved in the non-coding sequences in a messenger RNA that we call the introns. So eukaryotic genes are not continuous. The actual coding sequences are only found in the exons, the exons, which is the expressed, X is for expressed portion of DNA. And the introns, the N stands for in-between sequences, is what is not coded and what is actually spliced out of the messenger RNA during that processing period. In reality, eukaryotic RNA is only about 10% of the actual gene. So what that means is that the exon that is present in the messenger RNA is really only about 10% of the messenger RNA. About 90% of what gets transcribed is actually not the gene. It's actually this intron portion. So what occurs is a splicing out of these introns, and that's called post-transcriptional processing. So the primary transcript is this pre-messenger RNA that gets transcribed, and then the splicing occurs by an editing process that then makes a mature messenger RNA. This is a picture showing a little bit of how that process looks. And we're going to look at that a little bit more carefully, in particular the role of a spliceosome in cutting out these introns. But you can see the spliceosome, oh my goodness, what just happened here? The spliceosome is a large molecule, and it actually has some different types of RNAs. There are SM RNAs, which are small RNAs that are found within this spliceosome. And within this complex, there's a looping that occurs where there's actual cutting out of the intron and also a rejoining of the two exon portions on the RNA molecule. It's important that splicing is accurate. There can be no room for mistakes because, as we've talked about and as you learned in regular biology, even a single base added or lost will throw off the reading frame that is going to be responsible for the different codons making the sequence of amino acids in the proteins. So ribozymes are catalytic RNA molecules that function as enzymes, and they can splice the RNA. And then we have these small RNAs, the SN RNAs, which are actually small nuclear RNAs that are involved in this splicing action. And these combined with proteins make up this large molecule that's called a spliceosome. So again, there are special small nuclear RNAs that are bound together with some other proteins to make this spliceosome. And this is the action again that we get in pulling out the intron. You'll have two of these spliceosomes joined together, or two groupings actually in this picture of snRNAs that then together will make up this spliceosome complex that is able to pull out this excised intron. A spliceosome is usually made up of about 5 snRNAs in over 150 different proteins. And these snRNAs, when you bring them together into a complex like this, they actually become snRNPs, which we actually pronounce that as SNRPs. And these are called small nuclear ribonucleoproteins because they actually are not only the snRNA, but there are actually other proteins involved in this little complex that's made here. So these SNRPs then come together and help make up the spliceosome. One of the things in the processing of RNA is that we can end up with different types of messenger RNA depending on how the splicing occurs. So what they've actually discovered in recent years is that we get alternative splicing, which means that different portions of exons get cut out or different portions of introns get cut out so that we can actually, with one messenger RNA molecule or transcript here, we can end up with 2 different messenger RNAs that have just been actually spliced in different ways. So the last portion, again before the messenger RNA that has had its introns taken out, is to put on that 5' cap and that poly A tail. So a longer tail means that the messenger RNA will last longer, which means that it will be able to be read by the ribosome more often and produce more proteins before it degrades. These modifications, this addition of the cap and the tail, actually seems to result in several or serve several functions. It helps to facilitate the export of the messenger RNA to the cytoplasm. Its job is to protect the messenger RNA from hydrolytic enzymes that are located in the cytoplasm. And they also help the ribosomes attach to the 5' end. So this cap is important for the attachment of the ribosome. And I'll end this section here with this picture, which is showing the addition of the 5' cap and the polyadenine tail.